pattern of migration, and using two different types of gels. One of them is the agarose gel, and the other one is the SCS gel. And so there are differences between both of them, and I'm going to go over them. But before that, I'm going to start talking about serum proteins. And so do you guys know, so you guys remember what the definition of serum is? So does everyone, when we're talking about serum or sera, what are we talking about? What would be the definition of serum? What kind of things did you expect to see in serum or in serum? So anybody?
Proteins.
You can see proteins. What kind of proteins?
Transfer.
Sorry?
Albumin and globulin.
Yeah, albumin. Yeah, so albumin. So albumin is a big one. So the immune proteins, right? Which is what do we call them? Okay, so albumin, and so the other things that we will see were the IgGs, right? So I misspelled albumin. And the IgGs, and there's something called ferredoxin and some other stuff. Okay, so if you look at the actual distribution of serum proteins, so remember the serum proteins, would you expect to see cells in the serum? Do you have any cells? Do you have any clotting factors?
No.
Yeah, that's correct. So you wouldn't see expect to see any cells because those are removed. And so by definition, sera is when you take out all the cells, the red blood cells, the white blood cells, and you also take out the clotting factors. And so the only thing left is serum, right? So this is a liquid with a lot of proteins in there. And so when you look at an analysis of the proteins, one of the main ones, like we mentioned before, is albumin. So if you look here at this table and you look at the grams per liter, so I'm only going to look at this side. Albumin by far is the most abundant, right? So it's almost 50 grams per liter. And then the other ones are distributed. So the globulin, so like gamma globulin, and all of these are also found in there. So all of these are proteins, part of the immune system. The gamma globulin. So you see that the IgG, the gamma globulin, is also very abundant. And so if you look at the distribution in terms of like percent. What you see is if you put them all in a chart, like a pie chart, like this, what you can see here is albumin takes almost half of the space or the concentration of these proteins. And then you have IgG, so IgG is very abundant. And then you have other ones like transferrin, the and transport proteins. You have fibrinogen, so fibrinogen is involved in clotting. So this is like in wounds when you make a clot. So these are not the clotting factors, so this is a protein. And then of course you have the IgM. So all of these are in smaller quantities, but they're all important. Because you have something called complement. So we don't talk about complement here, but does anybody know why we have so much albumin? Why do we need to have so much albumin in the blood? So it's almost 50% albumin just by looking at this chart here. So why do we need so much albumin? Does anybody know? What does albumin contribute to that? So you have essential things in the blood. You have white blood cells for the immune system. You have red blood cells that deliver they deliver oxygen. But in terms of the proteins, why is albumin so abundant? You guys have any idea of why?
Is it because we did talk about a lot of things last time? I don't know if it has to do with albumin. It doesn't have anything to do with the clotting factors, but we talked about like the pressure of the cell, right?
Yeah. So the osmolarity, right? So it has to do with osmolarity. Yeah. So it is essential for how things are, how the blood works actually. Even though it doesn't have like an immune response, it doesn't deliver oxygen, but it helps keep the pressure inside the the blood goes through. When you think of the blood vessels, these are tubes. So these are vessels they deliver oxygen, right, and deliver food, nutrients. And so, but the function of the albumin it has to do with the osmolarity. So it keeps the osmolarity inside in the blood, in the blood. It keeps it at a high osmolarity. And so the reason is because remember the blood coming out of the arteries has a very high pressure. So I don't know if I remember exactly how much I cover out of this, but I'll draw it in I'll try to make a blood vessel. So you can think of it the heart here. So the heart is a muscle, right? And it's contracting, right? And it's pushing the blood, oxygenated blood out of the heart, and it's sending it to the tissues. So you have this heart that's pushing the blood, and then you have the aorta here. So the aorta is a big vessel in a blood vessel, so an artery. And so these aorta, the pressure coming out of here, the pressure is very, very high in here from the contraction of the heart is pushing the blood in one direction. And so either it goes to the upper extremities, to your arms, or to your head, to your brain, but it also goes to lower extremities. So down here, where the lower extremities are, so just pretend these are the lower extremities. And there are vessels or arteries, small arteries coming out of here. But let's just say, for simplicity, that's only one vessel. And so the pressure here is high, it's very high, and so that is needed to basically to push the fluids outside through the membrane to the blood vessels into the tissues, right? So you deliver a lot of it is water, but a lot of it is oxygen. And so the water goes here, is pushed by this pressure from the aorta. Because remember, the blood vessels, the aorta, or the arteries, they're very elastic, and they can contract, and they can muscle, smooth muscle around them, and they can contract, and they can stand the pressure of the coming out of the aorta. But the pressure at the same time is what is a force that delivers the water, right? Some of the water into the tissues. And so what happens is you will lose a lot of water, and if there was nothing coming back, if you lose a lot of water over here. If you don't have another source of pressure or another something that will bring the water back into the arteries, and so this pressure it's the so that pressure here that you started over here is lower over here. And so now there's a different kind of pressure here, and this is created by albumin. So you can think of albumin as this a lot of these proteins in here. So what the albumin does is it creates a difference in water between the vessel and the tissue here. And so that's pressure is called osmotic pressure. And so the albumin basically attracts the water back to the blood vessel. And so this is a different pressure. This is osmolarity. Over here, the pressure is more like a physical pressure. Okay, so the albumin is important to bring back the water inside to the arteries, and so that's I mean yeah into the arteries, and so that's why there's always this force between the osmolarity and the physical pressure from the pumping of the heart, and so one delivers water, but then the other one brings it back in. So the pressure here is highest is due to the osmotic pressure. Okay, so this osmotic pressure is created by the albumin here, the concentration of albumin. And so if the albumin concentration here was very low, then the person will lose a lot of water all the time to the tissues. It will be clear, but then the pressure in the blood will be very low. Okay, so now let's go back to second. Here it is. Okay, so now we know about why we have so much albumin. Okay, so one of the things that you analyze serum proteins in a clinical setting, so you can or in research is because the all these proteins, the concentrations, can tell you something about the condition of the person, the physical condition. And so some of the things they can tell you is if you have high values or high concentrations of these serum proteins. And so, for instance, if there is a lot of albumin inside the concentration, that means the person is dehydrated. And, and so dehydration, of course, can be dangerous for long periods of time. And then you have all the alpha 1, the alpha 2. A lot of these has to do with inflammation. Sometimes it detects it has to do with infection, kidney disease, in terms of in high amounts of alpha 2. And so some of them had to do with cholesterol. Okay, but the one that they look at more is the IgGs. And so because the amounts of IgGs, if a person has really high amounts of IgG, that can suggest that the usually it's not good, that the person has a type of cancer called myeloma. And so this myeloma, it's you can detect it just by running a gel, because the IgGs they migrate different. Okay, so they're in higher concentration. So when you run them on a gel, they're gonna be the concentration of the fractions are gonna be much higher, and also the way they migrate to the gel is gonna be different. Okay, so in a lab setting, the high IgGs, it can tell you if a person has myeloma. At least there's at least two types of cancer that can be detected by high IgGs. So this is cancer of the cells that produces the IgGs, and so the person that is diagnosed with this cancer, they produce like 10 to 100 times more, it's really significant high amounts of this protein. And if you know a little bit about the kidney, so the kidney filters proteins, and so the kidney eventually cannot support filtering all these IgGs, all these high concentrations of IgGs. So a lot of these people die because of kidney failure. So myeloma patients. So it takes time for these conditions to appear, and so once they detect them, it's usually because there's already a lot of IgGs present in the serum of the patient. And so There are a few conditions where the concentrations or the values of these serum proteins are low. Some of them have to do with the opposite, is the gamma globulin, if it is low concentration, then it has to do something with some condition related to the immune system. Or people that have poor nutrition, so who have low amounts of these IgGs. In some cases, there's conditions, some type of thyroid problems or liver disease, and again, inflammation in general. So those are um, some of the conditions that can lead to lower serum proteins. And so one way to analyze them is you can actually run the gels of or the patients or even animals to see how these proteins are distributed, the amounts. And so you will run a gel, and so you will get a pattern, right, of fractions. And depending on the gel, you get the fractions representing the different proteins. And so one of the gels or is the albumin will show up here at the bottom. And so they're moving in these directions, the proteins. And the other one could be IgGs. It could be other things like transferrin. Okay, so in the one thing that people can do is they can actually look at the thickness of the band, and they can relate the thickness and the intensity. And so this is a lot more intense than this one right here. And so they can actually use a piece of equipment called a densitometer, and it will give you something like this. So it will give you the amount, the relative amounts or specific amounts if you have something to compare with. And so let's say this is albumin, and relatively to the other ones, or you can know that you can run a known concentration of a protein, and then so you can see that the IgGs, whichever you are looking at, is going to run, and so these are amounts. So these are amounts. So you're gonna basically what you get is you're going to get a I'm not writing here very well. Okay, so this is going to be the amounts or concentration. Okay, so concentration here. This is a sign for concentration. Okay, so what you get then is you run a gel and you get something like what I'm going to show you next. Okay, so you're gonna get something like this, and so you can see here this will be using what is an agarose gel. And I'll tell you, I think we talked about this before, but I'll talk about it a little more. Is this is where you run the gel in using an agarose gel in a pH of 8.6 buffer that makes all the proteins negative. So pH of 8.6. And so what you get is you get something like this. You get you get a pattern like this, and this is the relative amounts. And also it tells you which ones come first on the gel. So the albumin is the one that comes here at the bottom, moving to this side, and then the gamma is going to be over here. Okay, so by running the gel and then looking at it this way, so will be a normal person. So this would be a normal, and then you can compare to a patient. So you compare to a patient, and so you run the same type of gel, and so a person that has this myeloma, the pattern of migration or the amounts from the analysis is going to be like this. And so what you see here, which one is more abundant now than the normal? which protein. You can see this graph. So you see this is albumin. So which one is more prevalent now, or where the concentration is higher than the normal person?
Gamma.
It's in gamma. It's in
The gamma, right? So the IgG. So just by looking at this gel, you can tell that this poor person or patient probably more than likely has myeloma. And so the person originally doesn't know, of course, or the doctor, is just because of the symptoms of being tired or having kidney issues that they need to running gels like this, or the analysis done in the labs. So here again, you see the same pattern. You see the albumin, you see the alpha, and then the beta, and then you see the gamma that is really, really high. That's because when you look at the actual gel, if this is the gel and moving in this direction, if you have the albumin here at the bottom and it's very thick, the gamma here is also going to be very thick. And it might be now the fraction might be migrate also differently. But you can see it now. So this would be the patient here. And then the person that has a normal IgG, the fraction may only look like it's like a very single line or a very clear, very short line, and then the albumin will be the same amounts. Okay? So this is here they show you an actual gel. So you will see here, and then the gel is shown here next to the fraction. So the albumin here is 60% of the total protein. The gamma chain is also is 16%, but it runs higher. So this is from going from right to left. But this is the class, so this is the, so remember you're running with a pH, if you're running in a buffer that is, they have some electrical current, so everything is migrating to the positive side. And so all the proteins are charged. So you normally will see them from the negative here on top and the positive over here. So all the proteins, and so this is an agarose gel, and all the proteins are migrating by charge, by the amount of charge that they have. And in this pH of 8.6, I think it was 8.6, 8.6, all the proteins are negative, but because albumin has a lower pI, so albumin, and I don't remember because I don't memorize the PI. Albumin has a lower PI than the IgGs. Okay, so IgG is a higher PI. So for this reason, this albumin is more negative than the IgG, and so the albumin migrates faster because it's moving to the positive charge. So it's more negative, so this will be albumin here. So I'm just going to write Al for albumin. And then the IgG, so the ferredoxin of the other proteins, they were going to be over here. Okay, so this is IgG. So this only happens if the pH is higher than the pI of these two molecules. Okay, any questions?
I guess I do have a question.
Yeah.
So I know that you're explaining this to us and based off of the PowerPoint. So for example, if you were to test us on something like this, what would we expect to see like on a quiz or like on our midterm? Like how would we expect the question to be worded and what would you expect us to do?
Okay. So the way it will be worded, it could be something like this. I could be you. I can tell you. So we're running a sample in an agarose gel using. I may or may not tell you the pH. So I won't. It depends on what I'm asking you, but you basically need to understand the. Like if I tell you one protocol, you need to understand what are the conditions and why it works that way, and how the proteins react under those conditions. So in an agarose gel, I'm telling you the pH is 8.6, and it makes all the proteins negative, right? And so you're supposed to understand why are the proteins negative is because of the PI, and the buffer is higher than the PI. And so the reason albumin migrates faster, so albumin migrates faster is because it has a more it has more of a negative charge. So you're supposed to know in an agarose gel they run by charge, right? So they run by charge. And then the next gel that I'm going to talk to you about, they're going to run by size or molecular weight. So here you, but you're supposed to know they're running by charge, not by size, and what gives them the negative charge. So you need to understand that they're negative. The proteins are negative charge, and what gives them the negative charge is the high pH that is higher than the PI, and also the PI is lower than 8.6. And so you expect to know don't expect you to memorize where all these bands are, but at least two of them, at least the albumin and the IgG. Those are the kind of like the most important, right? In terms, the IgG tells you where everything is going. It kind of gives you it kind of tells you what kind of gel you're running as well. And so those are the things. So I don't want you to know the pattern of these, but do recognize that this is a normal person, right? So if I tell you this is an agarose gel, and this is, you might have to say, well, this is a normal person. And so what about the other one? This one, you can say, so here you know this is what type of condition does this gel tell you, or this graph tells you, based on what we went over. What type of condition do you have here, or what kind of gel will give you this graph? Okay, so then here you expect to say that this person probably has myeloma. You can get all kinds of conditions, but I don't want you to know these conditions. I don't want you to memorize them. All I'm telling you is that the concentration of these proteins change, then can you get some type of medical condition. So like this one has to do with inflammation. These two guys here, alpha 1 and alpha 2, they're elevated. But you don't need to know these. You don't need to memorize. This is just to show you. This person here has cirrhosis. The IgGs run not elevated, slightly elevated, but they run at a wider um, uh, pattern, probably because the IgGs are altered in some form. This one here has myeloma here. So this one you know because of the IgG. And then this one has something called nephrotic syndrome. Does anybody know what the word nephrotic means? You see the word nephrotic. You know what a nephron is?
Nephrotic has to do with your kidneys.
The kidney, correct. Yeah, the kidney. So this poor person has the kidney disease. Okay, so this is just to show you that In the labs, if you run these two type of gels, you can get you can show you different things about possible medical condition that the person might have. But no, I don't want you to memorize any of these. It's more to understand it. Okay, so that's a good point because we do have a quiz next Monday.
So I know that you're saying that you don't want us to memorize these, and if you don't want us to memorize them, okay, so I guess the question is like, whenever you're providing us a question, you're going to be telling us like, okay, this is where this is where there's an issue. Like you'll tell us like whether if it's in the albumin or if it's in the gamma. And then we're supposed to know, okay, like if there's an issue here, this is probably or possibly what it could be. If there's an issue here, this is probably and possibly what it could be.
Right, yeah, or I can show you a gel and then you could, or a pattern like this, and then you tell me yeah, there's a problem here because this doesn't look normal. And so I may not ask you about the disease, but I may ask, you might say it's cancer, more than likely myeloma. But yeah, no, I'll give you some details. Nothing to memorize, nothing other than, the understand the process, the concepts.
I see. Okay.
Yes, but no memorization. So yeah, no, I don't believe in memorizing. I don't like to memorize things. I like to understand more. And so there's a few things that you do, kind of memorize. So, but like, the name myeloma, but almost everybody knows or heard. But you do need to understand, like here we talking about two different kinds of gels. One is an agarose gel, and that I've been talking about, and I think I went over it last time also. An agarose gel, and the other one is the SDS page. Because the proteins look completely different and the conditions that you run them are different. And so you basically need to understand what you get out of these, how you run them, and what are the ingredients that gives you the results that you expect, that you are seeing. So for instance, here, the most critical thing about the agarose gel is the pH. And so you understand that the pH it's a high pH. There's a reason why there's a high pH is because it's going to make the proteins, it's going to give them a negative charge. So you understand that, then you'll know everything about the agarose gel. The other thing you need to know about the agarose gel is how they run. They run the albumin runs faster than the IgG, right? So like I show you. And so for albumin, yeah, if there's something wrong with the albumin or the amounts, then you can say, you don't need to know the name of the disease, but you can say, oh, this person has a condition related to the amount of albumin. And so it might be related with osmolarity. Okay, so this is the thing about the albumin, the agarose gel. So, and then I'll go over the other one, the SDS page, and I'll tell you what are the important things to know about the SDS gel once I go over it, the actual protocol. Make sense? So another thing that you can do with the serum proteins is you can analyze them using something called the chemiluminescence, which we don't do here, and I'm not going to go over them. But all this is just to tell you that there are conditions, like in this case, where the IgGs increase, not just in myeloma. But it's also in some viruses. So in this paper, I'm not going to go over the paper, I'm just going to go over some of the results. And they're looking at amounts of three different IgGs. So they're looking at IgG A. So here is IgG A, IgG-M, so M, and then IgG, right? So the main immunoglobulin or antibody IgG here. And they're looking at levels of these antibodies in people that have, and so I didn't read this paper very well, but just looking at the data, the results, they're looking at these levels of antibodies that people that were they were diagnosed or they had some affected by diagnosed, so they were exposed, not exposed, but they got COVID. And so they're looking here at mild cases, moderate cases of the disease. And when I mean moderate cases, is severe. So most of the people that get affected or infected by the virus, most of them are mild or moderate. It's only a small percent that get severe and then end up in the hospital or die. But they're looking here at the antibodies. And so what you see here, and by just looking at what antibodies increased during this viral infection. And so you can see here, and they don't tell you how long after the well, that's probably because I didn't read it carefully. So they how long after the infection they looked at these levels. But just looking at IgA, you can see here that these severe cases, the IgG levels went a little bit higher, and moderate levels are somewhat higher, and then mild levels, the IgGs didn't even respond here. So that makes sense because the person really didn't get sick. And so the levels are going like this. This is IgA. And in here, this is a little different. So this is IgM. Okay, so this is IgM. And you can see here, in severe cases, they didn't increase too much. Mild cases, they didn't increase at all. And then for some reason, in moderate cases, they have a little higher levels of IgMs. And so, does anybody remember when IgMs act, when they are important? So did you remember? So what do you remember about IgM? Anybody? Anything about the structure or when is the onset of when they work? So IgM is a pentamer, remember? So IgM is a pentamer. And they tend to work early in infection. So the other one, though, the IgGs, they in mild cases they can go up, but in moderate and severe cases they go higher. Okay, so the IgGs, and that makes sense because you get infected, and eventually you get a response from the antibodies producing cells that later, if they get reinfected, then we have the antibodies to fight the disease, right? And so again, this, I don't want you to learn this. I'm just showing you to know that the IgG's levels increase, not just when you have an inflammation, but also when you have a viral infection. And so this is the natural response of the immune system. Okay, so the next thing is the actual gel. How does the actual gel looks like? So these are beautiful gels. They're looking at so this is agarose gels. Agarose. Okay, so what are we looking at here? So what are we? How are we distinguishing these different fractions? How are we? What's the? Why are they moving in this way? What gives them these characteristics? Okay, so again, albumin here, this is the normal healthy person. Albumin, this is the positive side, this is the negative side. Albumin is the most abundant here. This is what you expect, right? And then you get these fractions here. This over here is the gamma, and so these are moving by positive charge, right? So how are these proteins moving by? What's affecting them? What's the characteristics that makes them move in this direction? Anybody?
The size?
No, they're not moving by size. Remember I told you about by charge. Yeah, so they're moving by charge. Okay, so you remember this. So anything I ask you about agarose, you remember these and the pH, PI, and then this is albumin, then you do well on this part of it, or the test or the quiz. Okay, so this is an agarose gel. The odor here, the one here on the right, this is the one that has myeloma here. As you can see here, everything runs almost the same, except the gamma globulin, it's increased in concentration here. So it also, you can see here, it runs different. So the pattern of migration is different. But the most important thing is the amount. So the amounts of gamma globulin in the myeloma patients increase. So the concentration is highly elevated. So this is how you would distinguish from a person that has myeloma and a person that is healthy. You see here that you don't see any bands here. And so the albumin is the same amounts. Okay, so you will say here the person has myeloma. If for any reason you see a gel that all the amount of albumin is lower than the normal, but everything else is the same, then the problem with this person is going to be some condition affecting the albumin, which we don't need to know any names, but it just. So just remember, this is the pattern that you get when you run an agarose gel. So here, this is globulin and this is albumin. This is what I didn't ask you to read the actual, the protocol that we use in the lab. And this is one of the pictures they show you in their, the company that sends you it's a picture of how the different fractions migrate. So here again, this is albumin. This is alpha 1, alpha 2, beta, and this will be the gamma fraction here. It's just showing you these are called the globulins. This is albumin. Okay, so that's all we need to know about this gel. Okay, so another thing I need to understand about gel electrophoresis. So this is agarose gel that I've been talking about. We ran this in the lab last year. This is what you will be doing if we were in the lab. You will be running a gel, an agarose gel. And one thing about the agarose gels is the proteins remain native. Does anybody know what a native protein is? So you remember from biochemistry, you took biochemistry. What is a native protein? Or you heard something in biology. What is native protein?
Would it be something related to like a similar protein or something like that? Or
A cellular protein. So it has to do with the structure of the protein.
Wouldn't it be at their regular state, their natural state?
Yeah, the natural state. Very good. So yeah, so remember proteins, they have the primary structure, the secondary structure. The primary structure is the actual sequence of amino acids, right? But then the needed structure is a three-dimensional structure that the protein, when it functions, when it's natural, the three-dimensional structure of the protein. So I could have a protein that looks like this soccer ball here, right? So this is the total structure of the protein, but three-dimensional. This will be called the native protein. This is the functional protein. It has to be spherical in order to work. However, if you disrupt all the bonds, remember proteins are peptides, right? And then the peptides interact with each other, so they can form bonds in parts of the protein. And these bonds is what gives them the third dimensional structures, right? And they have all kinds of ways to interact. You can have hydrophobic bonds between different peptides. And so eventually they get to the whole thing, it gets to a three-dimensional structure. However, if you treat it with high acid or pH, or you denature the protein with an acid or a base, or with heat, you unfold these, you break all these bonds. So you break them, you denature, you break all the bonds, and then the protein is going to be unfolded, right? You can think of it just like a long spaghetti, unfolded. And so in this case, the protein is denatured, is no longer native, is no longer normal. So this denatured protein is one that lost function, so no function. And so the same can happen to the albumin, to the IgG, the antibodies. They can lose their shape, or they can become denatured. And one way to denature them is to boil them or a detergent. Okay, so the advantage of using these agarose gel is that they retain their native structure for the most part, because you're not treating them under harsh conditions. Even though the pH is a little higher, it's close to 7. So so the separation here, like I already told you, is by charge. Okay, so the more negative, the more faster they're going to move. Okay, so that once the gels, so this is what we were supposed to do last year. So we run an agarose gel of different sera from different animals. And then you stain the gels with something called proteins. Usually you stain them with Kumasi blue. Okay, so now, so this is what we were going to do in the lab. And so what I'm going to show you next is in a gel from a group from last year or the year before. Okay, so here, what you have here is this is the agarose gel, and this is the serum that they loaded here. Okay, so they loaded the serum, and so just for analysis, they to compare, I asked them to. So this is cow serum here. This one here, serum from cow, and then I asked them to load albumin. We have albumin in the lab, so just purified albumin. So as you can see here, this is albumin. So that makes sense, right? Because it's an agarose gel. The first band that you see is albumin. And so the serum that the students ran also has albumin. So that's good. And we were running these to make sure to run them in the lab, but also because later on we separated the IgGs from albumin. And so the other thing they ran here, so the other things you can see very well, but it's a gamma globulin. So, and I think it's this one right here. Or it might be this one, I'm not sure. But the thing is that the globulins ran higher, the IgGs. So this is what you would have gotten, something similar to this. And so this is just, again, because later on they use this agarose to purify the IgG. And Later on, they were looking to see if the IgG, so they purified them by going to a column. If the IgG that they purified was pure and didn't have any of the other proteins. And so in this case, this is very clean, but we're not talking about purification right now. Just listen out, just running, we were just running the gel here. Okay, so what can you see here is if you look at this gel compared to, so this is what the students ran, compared to the one I showed you before. What can you tell the difference? So this is in a clinical setting. This is what the gel looks like. In a real lab setting, this is what the students got here. So what can you tell about this gel? Can you see the fractions clearly?
No, you can't see it as
I
clearly.
feel like, I don't know, like I feel like you can't really differentiate. It just looks like one really long line. Like you can't really differentiate the fragment
Yeah.
from each
So
other.
which is the only one you can really tell that is there?
The albumin.
Well,
Yeah,
I see four different things, but like the wells are labeled, like it says lane 1, lane 2, lane 3,
Yeah,
lane 4, but
yeah. So they're not labeled. They're not labeled well. So they sometimes they could mix a well, and so that's why I didn't know which one is which one. I'm assuming this is the IgG. Okay, but the main point here was this one, and also that the albumin you can see is here. Okay, so this here, when you cannot see this, this is called a smear. Okay, so you have a well and you cannot distinguish the fractions. This is called a smear. And so at least you can see the albumin here. Okay, so when you have a smear, there are different reasons why the protein smear. Can anybody know why? Does anybody have any idea? You don't have short bands. And so like the student, if you get this in a gel, you don't get points off or anything. I just want you to understand that what could have happened, but not you won't get points off for this is something that can happen to anybody. And so, but the reasons are this is what I want you to understand. What are the reasons that you could have gotten this instead of uh, short, short bands, right? So we didn't get the short bands like what you see in the other gels. But what are the reasons you think might be? Uh, and I know some of you may not be been in the lab or worked with proteins before, but what are you just from looking at it? What do you think could have happened? It created this. A smear just means that the proteins are either so the proteins are degrading. So proteins are degrading. The proteins are of different sizes. So they could it could be that so it could be nothing wrong with the student, not the conditions that you did, but it could be that the syrup that you get from the refrigerator might be old. So proteins degrade over time. That's why when you buy fish, you buy meat. Not only do they can grow bacteria, but they can over time proteins degrade. So you can see a degradation of proteins, protein degradation, which the other thing that could happen is if you overload here, so you can overload the system. So these gels are very tiny, right? So you add too much. So you overload with too much concentration here. And the syrup would have been old. The other thing is if you run the gel, so we had to run them at like 100 volts. If you run them too high because you want to go home, you want run them at 200 volts, 120 volts. When you run the proteins very and the very high voltage, they just go faster. Also, the other thing is the agarose itself. So we made the agarose. Uh, no. This is the technicians made the agarose. So I'm not blaming the technicians, but it's possible that the concentration of the agarose might be, might not be what you wanted, the right concentration. But more than likely could be a combination of these two factors, degradation, overloading, or running the gels too fast. And again, it could have just been the sera that we had in the lab was old. But in any case, this is what a lot of you would have gotten. You would have gotten some of you would have gotten uh, better resolution than others, but this is just one example of what I got last year. And so the thing though is that we were using these later to purify. So we know we have IgGs because later on we purified them and then we got a short band. Okay, we were able to quantitate it. Okay, so that's the agarose gel. So the next one, keep up. So that's the agarose gel. The other gel that we ran last year is called the SDS page gel. So this is some SDS. SDS stands for, sorry, getting ahead of myself. So SDS is a detergent. And so what happens is you add the detergent SDS, and then you also, another thing you do to a protein is you boil it. Okay, so you boil, then you add SDS. And this gel, we buy this gel. This is using a gel that it's more standardized because we buy it. So the bands tend to migrate better. That's another thing about when you run native gels, they don't run as well as gels that are denatured. But in this case, in the SDS page, this is the method called polyacrylamide. The polyacrylamide is synthetic. So it's more uniform than the ones made by the technician. I mean, there's no fault of the technician, of course. It's just that this is just the nature of the gels. And so this is a agarose gel here, but you see, I mean, an SDS gel, sorry. And the other thing you add here is, so you do three things to the protein. So the protein are going to be completely denatured. You add something called BME, or beta mercaptoethanol. And what this BME is doing is, remember, you have the antibodies, they have this structure, right? So you have this structure that has the long chain or the heavy chain. And then they have the little chain or the light chain. And all of these are connected by what kind of bonds? Does anybody remember? They're connected like this.
Disulfide bonds.
Yeah, the disulfide bonds. Yeah, very good. So these disulfide bonds. Okay, so these are going to be broken by the BME. So here. So you do all of these three things, you get proteins that are completely unfolded, and then they're separated. So you're going to have the heavy chain, and then you have the little chain. And they're going to migrate according to size here. So remember the previous one, they migrated according to charge, right? So now they're separated. And so what happens here, and so here you have a lot of wells, but you only need to look at one of them. So here the ladder, this one is the ladder. The ladder is our fractions that you know the size. And so here again, you're moving from this direction. So here are the wells, and you're moving to the positive size. And here they're moving by molecular weight. Okay, so molecular weight, meaning that on the bottom, they're going to be the small ones, the small proteins. And in the top, you're going to have the large proteins. Okay, so anything that is like a complex of proteins or a very large protein is going to be on the top. Does anybody know what a complex of proteins is? What's a complex? Anybody has any ideas? So a complex will be like when you have two proteins that are bound together or associated with each other. So let's say this is like hemoglobin. So let's say this is one protein, this is another protein, and they're stuck together or they work together, and so they're a complex. When you separate them, they're no longer a complex. And so these can run very high over here. So you see this here, they don't even move, and because they're so big. But in any case, the main point, some of the main points here is that they're moving by size, they're moving by molecular weight, like I said. They are they're denatured. So the proteins here are completely denatured, and they're not native anymore. These will be denatured. And so now let's look at how these compare to the, and so again, we're only looking at just, we don't want to go over all the proteins. We're going to look at, so what you see here is that the smallest, one of the smallest ones is here, is the light chain of IgG. So now we're not looking at IgG, we're looking at the chains. So this is the light chain. I'm going to write LC of IgG. And then here you have the heavy chain, so the HC for heavy chain. Then now you can see here albumin migrates higher. So albumin is this one. It's above the IgG. So here you're going to have albumin. And this is because they're migrating by size. So ALB for albumin. There are other proteins. This one is called transferrin. This is a protein involved in transport. But you don't need to know that for here. So anyway, so what you see here, they're migrating differently than before. Okay, so any questions about the SDS? No? Okay. So just remember. Okay, so this is the actual protocol that we did last year. So yeah, I allowed the students to run, given some idea, but they run. Well, some groups ran a few microliters and some double the amount of microliters, and they did it. I let them choose what they wanted, but I gave them a range. And then the other thing, we used the BME, denaturing BME. We boil for 1 minute, and we load into the gel. So the one thing about also is that the dye had the SDS. And so one thing about SDS, the detergent, is that if you have a protein, let's say the protein is highly folded, once you denature it by boiling in the SDS and BME, it becomes linear. The SDS makes this protein negative. And so regardless of the size of the protein is going to be surrounded by SDS charges. And so if the protein is small, it's also going to be negative. And so they carry a negative charge, and they're not native anymore. So when you run a gel, SDS gel, the little one here is going to migrate much faster. So this is the little one. This is the big one here. The big one is going to migrate much slower. So this is the little, this is the big. Okay, so remember here you're running by molecular weight. Okay, so now I'm going to show you a gel from one of the groups. I'm assuming it was a different group. I don't remember who did this. I just got it from one of the reports. And so here, what can you see here just by looking at it? What can you tell me about the fractions of it? Can you see more? Can you see less? Do they look better? Do they look? If you look at the amounts, which one do you think is albumin? Just by looking at the amounts.
It would be the one like at the bottom, right? The one that kind of looks kind of merged. It looks kind of like with
it's a very high concentration, right? So this is albumin actually. So this is albumin. But was this what you expect based on what I told you before?
No.
Not
No,
really.
did they overload the wells? Is that possible?
Yeah, they overloaded the wells, but it was also they were supposed to run different, right? Do you remember from the what I told you how what should have happened to the IgG? So I can tell you these are the IgGs here.
So what's in the first well is considered the IgG, but the second and third well, that's albumin.
Okay, so yeah, don't look at the wells here. So don't pay attention to this
Okay.
because this is purification steps. But just look at these two. So this is actual serum. Okay, so yeah, this is serum or serum. Right? And so yeah, don't look at the other fractions. Just looking at this fraction or this one. What was supposed to happen based on what I told you to the IgG? So this is remember, this is an SDS page. What did they do to this sample?
They denature, they boil the protein.
They boil it, right? So they boil and then they added what else?
The
Are you referring to the detergent?
BME
Yeah, the BME. So I'm talking about three things that they did, right? They boil, they added BME.
and the how do you say it? Detergent.
The SDS, the detergent, right? And so what happens here is they run it at very high. So this is the ladder. They're running like they're a complex. They're not running like they're separated. So which component here would you suspect didn't work or they didn't add it? So something happened here because remember something is supposed to, if you have a complex, something is supposed to separate this IgG.
They didn't boil it to denature it.
Yeah, that could have happened. The heat was not, they boiled it, but not very long, but not, but there's something critical here that didn't work, I think. Of the three components, what is critical to separate the, so you have the IgG complex.
Maybe it's missing the detergent because it didn't go very far. Like
It could be all of those two, but there's something critical that, remember what holds this together?
The sulfide bond.
Disulfide bond, yeah, very good.
denaturing.
So they could have boiled it, they could have added SDS. They either missed the BME or they didn't add enough. But there's something else that can happen to BME. That happens to a lot of foods. What could you think of? Maybe not their fault necessarily. So there's nothing wrong with this gel. I just want to tell you that this is not the way it's supposed to be. The results that we expected. And so they don't get points off or anything. I just want you to understand that something went wrong here, and it could have not been necessarily the student's fault. I'm not, again, not blaming anybody. Just understand what happened, what needed to happen that didn't happen. So the BME. So what could have happened to BME? Let's say they didn't forget to add it. So maybe they added very little. Yeah. So BME kind of smelly. It smells like skunk. Can you guys ever smell BME? So
No.
No.
one thing about BME is that I never told the students is that BME has a shelf life. And so one of the things about our lab is we don't know when, what was, maybe the BME was from the last semester or the year before. So I'm thinking in this case that the BME was just old, okay, and it was not working anymore. Now I know this from, being in the protein lab for many years, that I will bet that this is what happened. The BME just didn't work. And so you didn't get the smaller bands of IgG here. You were supposed to have the light chain and the heavy chains before the albumin. But everything here is fine, other than. And so here you can see this gel. They didn't run it long enough because when we do this lab, people, you start at 4, and people want to once you get to 5 o'clock, 5:30, everybody wants to leave. And so they asked me, "Oh, can I stop the gel?" Well, sure, yeah, stop the gel, but as long as you know how to analyze it, what you get, and so no problem with me. But anyway, so I just want you to know this is what you could have gotten last year. We had run it. And you might, if we go back in March, you might be able to run a gel like this when we purify it. So we take these samples later on. We take these samples and we purify them. And what you are supposed to get is a band like this. It's a clean band like this, and they did here. So this is what we were looking for. We want them to get this a pure band, but um, purify IgG. So yeah, there's nothing wrong with this gel. I just wanted to show you that it doesn't look like. So actually, both of them didn't look like they're supposed to look. But in any case, there's nothing wrong with them. It's just an analysis. So are there any questions? So I'm done for today. Unless you have any questions, you're going to have a quiz next week. So we haven't run any labs, but it's going to be based on, again, understanding these concepts. And if, so Wednesday is the class. If you have any questions, you can send me an email. You can, Thursdays, I'm pretty free Thursdays after 11. So if you ever want to meet on a Zoom meeting, we can schedule a Zoom meeting if you want to go over anything that you don't understand. And so, unless you guys have any questions, I'm done for today.
How are we gonna take the quiz? Do we have to log into Zoom or it just take it on Blackboard? And how is it gonna happen?
Yeah. Okay. So So you're gonna do it. So you're gonna do it online, of course. Not sure you're gonna do it. Let me see. You're gonna take it anytime that day. Make sense? So,
Yes.
so it's gonna be online. What do they call it? Online anytime. The day that we're supposed to meet. So we meet on Wednesdays, right?
Yes.
Yes.
Yeah, we're gonna meet online, and so you're gonna take the quiz. Yeah, anytime that day on Wednesday. Either in the morning, you can in the afternoon, you can take it. And if for some reason, let's say for some reason, because I'm a little forgetful sometimes, let's say I put the quiz too late, like I make it available at 1 p.m. instead of the morning. So I will leave it until the next, I will leave it at least 24 hours, but I'll send you an email too. And I will leave it at least 24 hours available. Okay?
What is the quiz gonna cover? Just the first three PowerPoints, or is
Yeah,
also the one?
no, just the first 3 PowerPoints.
Okay.
Any other questions? So remember, yeah, we do have a class, but if for some reason, let's say if I just, let's say if I am able to cover the material in the following class, then I mean I have a class, but I'll let you know when I need to look at the schedule more carefully. But at least plan for the quiz. Oh, somebody has a question. Ah, you didn't see online what? Oh, the PowerPoint. Oh yeah, yeah, it's there now.
Is it going to be time?
Yes, so it's gonna be time, but you'll have plenty of time.
Since we're asking if it's going to be timed, can you tell us about how many questions we should expect to see?
Ah, okay, so the quizzes usually are not a lot of questions, so you will probably get anything. So a lot of my quizzes are a combination, so let me show you. So combination of multiple choice, which tend to be easy, right, for the multiple choice. Um, Short answer questions. So short answer means that like one word. You don't have to write like a whole sentence or paragraph. Short answers and the so short answer questions. So I may ask you. So what component do you need to break the disulfide bonds or something like that? Then you say, oh, BME. Or what by what characteristics does the SDL's page separates the proteins, and you say by molecular weights, and or I may ask you what components did you need for the SDS page to work? The main components, the three main components to the nature of the cell, the protein, then you're going to say, oh, well, BME, you boil, and you add SDS. Or I can give you in a scenario. So these will be short answers. And then I can give you in a scenario where I give you an experiment that didn't work, and then you're going to tell me why it didn't work. But there's going to be usually short answers, not long answers. And, yeah, so no, you don't have to write a paragraph or and the most you will have to write is 1 or 2 words. And so I usually have about 15 for questions, usually 10 to 15 questions. So probably more like 15 questions. Okay. So,
I have a question.
yeah.
Are any of your recordings going to be available for review purposes?
The what, I'm sorry?
Your recordings.
My recordings. Oh, you mean I tape the class?
Right, do you?
I don't have them, but I can do that. Yeah, I can try to do. I can go in and you mean like go like a voice thread?
If you have a voice thread available, that would help. If you have your live Zoom available, that's also a plus.
Oh, okay. Mhm.
Just in case, because I mean, I'm pretty sure I'm not the only one, but might have missed a few notes here and there.
Oh,
So
okay, sure. Yeah.
whether or not if I can go back and catch some of your lecture, because
Yeah.
just in case I might be missing a few notes, and
Yeah.
it could be something that you said that would be, you know, an answer for the test or whatever the case may be. So
Okay, yeah. I'll do the what I'll do. You guys are familiar with voice threads, right? So it's like a video. Yeah, I have a slide and then I record something and we go to the next one. Yeah, I can do. I think actually I had one for one of the lectures. So I'll just put that in I will put that in your content. So let me write a note for me.
yeah,
Yeah,
because
I can do that. Mm-hmm.
it would really be a huge help with
Mm-hmm.
some because you did a lot of writing. I try to get most of it, but
Mm-hmm.
I'm lost in transition on some of it. So I'm just trying to find out if there's any way you can, you know, put some of your recordings or slides available
Yeah,
this
yeah.
weekend.
So what I'll do is I'll it might be a little shorter than the actual lecture, but it will be the more important things.
That would help. Thank you. That would really be a huge help.
Yeah, so I would do this weekend, and yeah, and so then for the next classes, I will give you actually short assignments. They will, help you with the practice, but you're hearing in the lecture. The reason I didn't have any assignments is because everything was very basic for the first three weeks.
Hmm.
And so it was better for me to go over them than just to give you a ton of material to read. But I can also post some of that material.
So wait. So make sure you're going to do like a little small assignment for us to do or to review.
Yeah. Like starting next week for the next section. Yeah, you will have something like a
Oh,
homework.
okay.
Yeah,
Okay.
yeah.
Okay. Cool.
Okay. Okay. Any other questions? Okay. So yeah, any suggestions that while we're here in the online because normally what we do is just go in the lab. I have in the labs I give very short introduction, no more than 10 minutes, and then we go and do the actual lab. And then sometimes I talk like what the students are running the gels, and then I go back but here because of the online, I have to go over everything that I can think of. And so yeah, I will record this if it helps you, anybody. Yeah, and that would be like optional to watch the voice thread. It's not going to be assigned. Okay.
Okay.
Would it help if I give you like a practice quiz?
Yeah.
Yeah. Okay. So that's something I can do too. A practice quiz. Okay. 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 So that will give you an idea of what to expect. And I won't grade it, but when you do a practice quiz, just make sure you don't look at your notes because that will tell you whether you understand something or not. I mean, you can look at them, try to answer them first without looking at the notes. Okay, any other questions? And again, there's something not clear, remember, we can always, I don't have set off office hours, but we can always meet on Zoom. And Thursdays and Fridays, so Thursdays and Fridays are pretty good for me, especially Thursday afternoon. So if you ever want to meet for a short time and go over something, we can always set up a meeting.
When is the quiz available?
So for practice, yeah, I'm gonna try
Well,
to
well, not the practice itself. The actual
the
quiz.
actual quiz. Oh, it's gonna be next Wednesday.
Live during class or?
You can take it anytime that Wednesday.
Oh, okay, cool. All right.
Okay, any other questions? Okay, so if there's no other questions, then I will see you next Wednesday.
Bye, Professor. Thank you.
Thank you,
Bye.
Professor. See you next Wednesday. See you next Wednesday.
See you next Wednesday. Have a great weekend.
Mhm.
You too. Thank you.
Bye.
Bye. Thank you.
Bye.